Hello everyone, welcome to another year of Nerve. And if this is your first time joining us, we are the Neural Engineering Research Venture, and our aim is to bring neuroscience to life in Africa and around the world. And we are Davi and Siobhan and myself. Siobhan and I will be your hosts for the evening, and you can look out for Davi in the comments. So since it's been a while since we've been online, we have a couple of very exciting announcements to share with everyone. So the first is the Deep Learning Indaba mentorship to strengthen the African machine learning community via the development of fundamental skills, has a program that matches members of the African AI community with mentors for short-term personalized interactions across a wide range of topic areas. So you are all encouraged to either join the excellent global network of mentors, or if you identify as part of the African machine learning community, please sign up as a mentee. And computational neuroscience is included in the scope of these sessions as well. Then we have the very exciting Neuromatch Academy for 2021 that is officially happening again. So please do save the dates from the 5th to the 23rd of July for Neuromatch. And there are also rumors of the Neuromatch Academy deep learning after the Neuromatch Academy. So yes, these will maybe include mini workshop sessions. So please show your interest here by pre-registering, and if you do have the capacity, there's also a call for volunteers. Then, also very exciting, InstaDeep is hiring for their research and apply teams in Cape Town, South Africa. So for those of you that don't know, InstaDeep is a learning company that develops cutting-edge artificial intelligence products and solutions for major global and local clients in Europe, the United States, the Middle East, and in Africa. Their focus is on developing enterprise software systems that solve existing problems across a range of industries using advanced machine learning, deep learning, and deep reinforcement learning. So please be sure to check these opportunities via the links that Siobhan is providing in the comments. Then also do add these links to your browser tabs to catch up to all the neuroscience events that are happening online. So Worldwide Neuro, of course, you can check out the event series following the first link there, and then also NeuroJobs, which includes study and work opportunities. Then also check out the ABC Meetup group that is based in Cape Town and also hosts or advertises regular neuroscience talks online. Cool. So Siobhan shared all of these links in the comments. Then we don't have a slide for this specifically, but if you are interested in joining a budding community focused at the intersection of healthcare and artificial intelligence in Africa specifically, please do join the conversation on Discord. And I think Siobhan also shared the link there. Yeah. Cool. So then, as usual, our house rules. Please do use respectful and inclusive language. So we do have a very, very big audience, so keep this in mind. And then for the Ask a Question feature, we do encourage everyone to post their questions online. You can see the button at the bottom of your screen there. And also remember to vote for your favorite questions throughout the talk. After the presentation, we will follow the presentation with our Q&A session, and then we will invite everyone on screen to ask their questions live. If you prefer not to ask your questions live, please do remember to add please ask, as we will assume that you will be coming on screen otherwise. And if you don't have a mic, don't worry about it. We will ask the questions on your behalf. But again, please remember to add please ask. And if your connection is too slow, or the feed is not loading properly, you can select the compatibility mode at the bottom of your screen, and there you can toggle the audio vision help button to a setting that is better for your location. But if your connection fails completely, the talk will be available to stream after the event. Okay, I will now hand over to Siobhan to introduce our very exciting speaker for the evening and our special event logo.
Thanks, Julianne. Hello, everyone. I'm very excited to be back online. And today we have a very special event celebrating the seminal work of Larry Abbott and Peter Dayan. Both are regarded as leaders in theoretical neuroscience, having literally written the book on it. Their book, titled Theoretical Neuroscience, Computational and Mathematical Modeling of Neural Systems, is the first comprehensive textbook in this field and is considered the standard text for students and researchers entering theoretical neuroscience. We are very fortunate today to welcome our speaker, Tim Vogels, who completed his doctoral studies under the supervision of Larry Abbott. Tim is currently leading the Vogels lab from their new home base in Austria, where the overarching aim of the lab's research is to investigate the building of models of neurons and neuronal networks that distill and rearticulate current knowledge of how nervous systems compute at a mechanistic level. Notably, Tim is also instrumental in the organization of Worldwide Neuro, the Neuromatch conferences, and the Ibro-Simons Computational Neuroscience in Bizo. Thank you very much for joining us this evening, and I will hand over to you, Tim.
All right. Presentation. Not quite sure on the. I'm going to do Larry and Peter proud, but you invited me, so it's your fault. All right. I guess I'll be sharing my screen to talk about codependent plasticity rules. And from this point on, I really can't see you guys anymore, neither your comments nor your expressions or anything else. So I'm really talking against the wall or against the screen. Lovely, lovely experience. But I hope that, yeah, I hope you'll take something from it, staring at your screens at home and at lab in these weird times. So, talking about interacting synapses that can stabilize learning and also stabilize neuronal dynamics. This work is very much only the work of Everton Agnes, who is a former postdoc of mine, who just started his own group at the University of Basel, and who is also in the audience, and I'm sure will start texting me hectically if I do or say anything wrong. So thank you, Everton. It's been lovely working with you for 4 years, and I'm quite bummed that you left the lab. And tried to prevent it, but hey, I wasn't very successful. So what you saw in the background, what you still see in the background is sort of an artistic rendering of a cortical slice with mostly pyramidal neurons in the background. What you can't see, but what I've brought into the front in other colors here are all the other neurons surrounding and embedding these pyramidal neurons, which are the majority of neurons in our cortices, with their inhibitory structures. And as such, there is 16 or 15 different types of inhibition in cortex that are playing with these excitatory neurons, and they make 140 different connection types between themselves and with the excitatory neurons in each layer. And it is that structure of 140 different types of connections that supports all function and behavior that we can display, or that we would like to display. And it's thought that the structure is largely built by developmental and also activity-dependent learning rules. So, yeah, making the structure from a set of rules that are inherent to all of these connections. It's been the focus of an intense intense study over the last 70 years. There are some 50,000 papers on synaptic plasticity on PubMed, starting with, or maybe with one of the most famous ones being the chapter by Hebb. But all of these rules have largely focused on excitatory connections, either connections between excitatory neurons or onto excitatory neurons, and a small minority has also focused on inhibitory connections. But it's fair to say that we still haven't fully understood even the rules that we're looking at, and the status quo is maybe best described in this figure, where you see a change in efficacy plotted as a function of the arrival of pre- and postsynaptic spikes at the location of the synapse on the x-axis. And this change in efficacy may be changed, or may be further modulated by previous history of firing in the pre or postsynaptic neuron, as well as the state of the network. But what is fair to say that these rules that we've discovered in the normal circumstance, or even in the best of circumstance, I suppose, cannot explain how cortex constructs itself, in that when you implement some of these rules to function in neural network models, they very quickly lead to the buildup of feedback activity or positive feedback mechanisms that will lead over a short time period to what could be understood as epileptic seizure, but they're really just synchronous high frequency events, as shown in this raster here, where we're plotting roughly 2 seconds for 100 units of that network, and we see that it starts off firing at relatively low rates, but soon escalates its firing rates when we have the excitatory synapses implementing a plasticity rule, as this famous spike-dependent plasticity rule by B and Poo. And before anyone gets grumpy, I should say there are notable exceptions to this. There are a few numerical papers, as well as a few analytical or theoretical papers that have looked at this, and they have found that there are rules that lead to stability. These rules typically are either very, very tightly orchestrated, so that the parameter regimes within which they work are very narrow, and they focus on special cases. So in the case of this paper here by Friedemann and again Everton, who published this in my postdoc lab actually in 2015, they have very specific sets of parameters for each learning rule that they implemented, and found that with those they were able to learn memories into a previously randomly connected spiking rule. So what all of these works have in common so far is that when they look at plasticity of a certain type of synapse, like for example this synapse, they will implement a rule which is to be followed by this synapse according to what its pre and postsynaptic activity is, and if you implement another synapse, or if you build another synapse that's plastic, you build another rule. Possibly this is an inhibitory synapse, this is an excitatory synapse, but there's really no interaction between these synapses other than through the postsynaptic firing that they, along with many other synapses, may or may not evoke. And so the question avails itself if synapses wouldn't be acting or interacting more directly with each other also in their rules, since they're usually pretty close together, and because also they often use or detect some of the same ions postsynaptically. And there is evidence for such codependent plasticity. We call this codependent plasticity. For example, here is a finding by Harvey and Svoboda, as well as Jesper Sjöström, that excitatory neurons interact with each other via voltage deflections or calcium deflections. So when you have a large amount of activity coming from one synapse, a second synapse will experience potentiation, even if its own stimuli weren't necessarily sufficient to evoke LTP alone. There's other papers that look at inhibition controlling excitatory plasticity. So when you block GABA, you show that excitatory plasticity all of a sudden is flipped. So what led to potentiation before leads to depotentiation or depression after a GABA block, and vice versa. There is some evidence that if you block NMDA receptors, you also abolish inhibitory plasticity. So meaning that excitatory synapses could control inhibitory synaptic plasticity. And finally, there's also some evidence that inhibitory synapses control other inhibitory synapses according to whether the ratio of excitatory and inhibitory inputs at that synapse is balanced or not. And we'll come back to most of these rules or findings a little later. All right, So that's all good. There's evidence for some sort of interdependence or codependence between excitatory and inhibitory synapses. How should we implement it if we were to model it? Everton has spent a large amount of time thinking about this, and what he came up with was a pretty simple function in which the efficacy change of the synapse is simply a function of pre and post synaptic firing profiles as before, but then additionally he had two currents, or two detectors of currents, I should say, for excitatory and inhibitory currents. So excitatory currents being largely NMDAs, or calcium currents, I suppose, and inhibitory currents coming from GABA, being chloride currents from the dendritic neighbors of the synapse in question. And then, of course, the more traditional pre- and post-synaptic spike trends will continue to play a role in this learning rule. Okay. All right. So, how would we implement something like this, or what does the rule actually look like? Well, we started with local spike-wise effects that look very much the same as what has been previously reported. So these are rules that are inspired by the earliest work of spike time-independent plasticity. And then we added some sort of neighborhood action that captured the effect of currents coming from other or being detected from other synapses in the vicinity of the synapse. For example, here, a change in synaptic efficacy on the y-axis as a function of excitatory currents that are detected when there is spiking that happens at the same time, I should say, as the spiking at the excitatory synapse in question, so pre, before, post. And then we added an additional heterosynaptic effect that also changed the excitatory synapse strength, but into the other direction. So when there was a lot of excitatory current arriving at the synapse in question, we allowed the synapse to decrease its efficacy by the logic that it may not want to further contribute to an already threatening or over-excitatory or over-stimulating set of synapses. And so the resulting function is one where excitatory currents in the vicinity, small excitatory currents in the vicinity are leading to an amplifying effect, but large excitatory currents lead to a weakening effect. And this balance point, this fixed point between where it flips from excitatory strengthening to excitatory weakening is governed essentially by the ratio of LTP inducing activity and heterosynaptic plasticity. Then we also have the efficacy of the synapse being governed by inhibitory currents that are detected at the synapse. Again, the efficacy on the y-axis, the efficacy change governed by inhibitory currents, such that when there's little inhibition detected, the efficacy changes will be big, but when there's a lot of inhibition, efficacy changes are essentially gated off, so that when inhibition is present, this synapse will not be able to strengthen at all. Those two functions combined together will then lead to a spike-wise plotting as before, but with many modulatory changes, so that you don't always get the same efficacy change for the same pre-post pairing, but according to whether inhibition is stronger than excitation or excitation is stronger than inhibition, you will get different amounts of weakening or strengthening for different combinations of pre and post. Okay. For the aficionada on the side, I already mentioned this set point here is governed by the balance between LTP effects and homeostatic effects in this equation. You can also see this here, where we just plot the ratio of LTP and heterosynaptic effects the x-axis and show that the excitatory currents that are affected by it are essentially changing in accordance to that ratio. And this is not, the same excited currents, by the way, are not affected by the starting point, so how strong the excitatory weights are to begin with. Okay, we're happy to report that this rule that Everton built reproduces some experimental data. This is not totally revolutionary, I should say. There's other rules that have reproduced this, so Claudia Klopath, if she's in the audience, will ask herself why I haven't put her citation on here, simply because I ran out of time. But ask me afterwards if you're interested in other rules that are reproducing this data. The voltage-dependent rule by Claudia Klopath for sure can do this. Showing what we're showing here is that if you choose a fixed pre-post pairing of either plus 10 milliseconds of pre-before post or 10 milliseconds pre-after post, you get a certain rate relationship, so how often you present these pre-post pairs in a given second. And you see here that the model can actually reproduce the changes in efficacy that are going up with increasing frequency quite nicely. And similarly, our model can also reproduce data in which pre and post are presented continually at 50 hertz stimulus, I think. So it's pre-post, pre-post, pre-post. And according to, if you have low depolarization, if you're far away from threshold, this will lead to depression, but if you're coming closer to threshold, then, or if you depolarize the synapses a little more, then this will eventually lead to facilitation. This is also something that our model replicates. In inhibitory synapses, the situation is similar, so we're implementing this kind of codependent inhibitory plasticity rule similarly, in that we start with local spike-wise effects, again, pre-post or post-before-pre, on the x-axis, and then the change in the inhibitory efficacy on the y-axis. This is a rule that may not look totally unfamiliar to some of you because it's been borrowed from a 2015 paper by Rob Frumke and James Damore, and also inspired by a paper that I published with Henning Spreckler and Wolfram Gerstner almost 10 years ago now. In addition to these spike-wise effects, again, we have neighborhood action. So when excitatory currents are detected in the absence of any additional inhibitory currents in the vicinity of our inhibitory synapse, then the synapse will experience strengthening that is proportional to the amount of excitatory current detected. When there is additional inhibitory currents, this efficacy change will be downregulated, such that for small excitatory currents, the synapse will actually weaken, and only for larger excitatory currents, the inhibitory synapse will still strengthen. And this will create, in essence, a set point that means that the inhibitory synapse will always try to balance the excitatory currents and get to the same amount of postsynaptic current. Similarly, inhibitory efficacy changes are also governed by inhibitory currents in the vicinity of the synapse here. When there is no excitatory current at all, then nothing happens, no changes happen either with or without inhibitory currents present. But if there are excitatory currents present as well in the neighborhood of the inhibitory synapse, the efficacy change will depend on the amount of inhibitory currents that are detected, and such that with small inhibitory currents you have positive changes, and with large inhibitory currents, again, you have negative changes, driving towards a situation in which excitatory and inhibitory currents are canceling each other out. So this rule will naturally have a set point that balances excitatory and inhibitory currents in the vicinity of that synapse. So this leads to a similarly changeable rule as before. It stays symmetric because we've made it so, but when you have more inhibition than excitation, then the inhibitory synapses are weakened. When you have more excitation than inhibition in the vicinity of the inhibitory synapse, then the inhibitory synapses, pardon me, are strengthened. So this means, again, driving towards a scenario in which excitation and inhibition are equally strong. So as inhibition grows stronger, the strengthening effect of the plasticity rule will diminish until they're even. Okay, so this is what the rule looks like in short. And together with excitatory codependent plasticity, these two rules play quite nicely together, as you will see. If you have independent plasticity rules, this cannot be said, so they typically don't play nice together, as I've already sort of hinted at in the introduction. This can be quite nicely shown when you change the rate set points for excitatory and inhibitory plasticity, as done here, where in an inhibitory rate set point of this inhibitory synaptic plasticity rule from 2011 is set at 6 Hz constantly, but over the 25 or 50 hours of the experiment, the set point of the excitatory synaptic plasticity goes from 6 up to 10 and then back down to 6 and then down further. And so you'll see that as long as the two rate points are adjusted to one another, the rate really nicely stays at 6 Hz as expected. Also, the ratio of excitatory and inhibitory currents detected at the soma of the neuron is quite balanced, and the excitatory and inhibitory weights onto this neuron are stable. But then if you unilaterally change the firing rate set point of the excitatory synaptic plasticity, you immediately enter an arms race where the excitatory synapses try to reach 10 Hz, the new firing set point, but inhibition freaks out and sits on top of that by increasing its weights as much as it can, leading to an explosion of both excitatory and inhibitory weight for the duration of this imbalance in rates to infinity, if you will. So this will keep increasing, bringing the EI ratio to its knees, not necessarily dominating the firing rate so much, so you can't detect what's going on inside the cell at the level of the firing rates, but surely enough there is something very wrong because these weights will simply go through the ceiling. When you bring the set point back to being coordinated, the system of course stabilizes again, but if the excitatory set point is lower than the inhibitory set point, you will achieve that, but the weights are essentially going down to near zero because inhibition will simply excuse itself and leave the room. So these are situations which are not necessarily biologically realistic, because that happens not only when the set points are separated by quite a few hertz, but it also happens if you have minuscule changes in the set point of the firing rates of excitatory and inhibitory learning. Okay, with codependent plasticity, this doesn't happen because I already told you that the inhibitory codependent rule here is not seeking a firing set point, but rather seeks to balance itself with the excitation, and that leaves the excitatory rate fixed point to set the postsynaptic firing rate on its own. So regardless of whether it's 6 or 10 or whatever, inhibition will not care much as long as excitatory and inhibitory currents are balanced. So the weights of the inhibition are going to be adjusted according to where the excitation goes towards, and that of course depends on the firing set point that we give, but there will be no arms race because they're fundamentally looking at different qualities. Okay, when you combine both excitatory and inhibitory plasticity, we find that they can reproduce a number of experiments. The first and maybe my favorite experiment to reproduce that we have now fully reproduced, so I don't know if there's anything left to pick on that bone, but in any case, this is a piece of data from Rob Fremke published in Nature in 2007, in which he showed that you could relearn a tuning curve. So this is normalized current on the y-axis and pure tone frequency played to a rat whose pyramidal neurons in A1 are being recorded. And you see that the rat has a preferred frequency, or that pyramidal neuron, I should say, in the A1 of that rat has a preferred frequency of 16 kilohertz. So the input currents are strongest for that pure tone, and it drops off for other pure tones. And then Rob presents a new tone at 4 kilohertz in the presence of an additional nucleus basalis stimulation here, which delivers a flood of acetylcholine to the auditory cortex of this rat. And he sees that the excitatory synaptic receptive field rapidly reorganizes itself such that the previously preferred frequency will now receive much less excitatory current, and the now preferred frequency, or the co-stimulated frequency of 4 kilohertz has now a peak excitatory current that was previously the preferred response for 16 kilohertz. Inhibition stays stable, at least for the first 30 minutes or so of the experiment, but then over 180 minutes of the experiment crawls over, so it adjusts its weights so that it balances excitation inhibition for every one of these pure tones that are being continuously played to the rat for these 180 minutes of the experiment. And so we've rebuilt this experiment before in a single integrated fire neuron. In fact, we've rebuilt this already for the 2011 paper, but back then we couldn't control the excitatory rule. We only controlled the inhibitory synaptic plasticity and kept the excitatory synapse static. And the question I maybe was asked most in talks that I gave about this paper was, why are you focusing on excitation, on inhibition? Shouldn't you be looking at excitatory plasticity here at least as well? We've done this now. So in this model, we have now excitatory and inhibitory plasticity that are both modulating excitatory and inhibitory strength, and that are codependent. So they will both not only listen to pre-post, but they will also take into consideration input currents in their vicinity. Of course, in a single compartment neuron, that vicinity is all the other synaptic currents combined. Okay, so the starting experiment is very much similar to what Rob sees, and in this situation where you have excitatory and inhibitory currents that are arriving at the same time, remember that excitatory efficacy changes are essentially blocked by this inhibitory function that doesn't allow or disallows excitatory changes simply in the presence of inhibitory currents in the vicinity. So whichever stimulus you give here through whichever pathway to that system, because excitation and inhibition are co-tuned, you will not see changes in weights. And this is the result that I just said, that I just described, plotted. So here on the x-axis is the time of the experiment, on the y-axis are inhibitory strengths and excitatory strengths plotted respectively according to this color scheme, where the red or pink here is the preferred frequency and the green is what's going to be the new preferred frequency. And the other two are some other weights that are, we'll just plot them alongside. So then for a short moment, we modulate the balance of excitation inhibition in favor of excitation. So we downregulate inhibitory efficacy, as was suggested to be the case from a acetylcholine stimulation in auditory cortex. So that changes the efficacies of both excitation inhibition, making the excitatory synaptic inputs more prominent postsynaptically. And that, of course, led to an immediate gating of excitatory plasticity by way of turning off inhibitory currents. and allowing excitatory changes, which then led to the rapid development of this new preferred frequency in green. And initially nothing much happens in the inhibitory synaptic part, but over time, in this case over 400 minutes, the inhibitory synapses will slowly adapt themselves to the correct value that would then balance excitation inhibition, and that's what we find at the end of the experiment. Again, we see that excitation inhibition are co-balanced. This is a result that's been published before for the inhibitory synapses. What's new here is that excitation inhibition will not get into each other's hair, but rather collaborate quite nicely in learning a new memory trace and then maintaining it. So here again, model and experiment side by side, and this may not look super cool for the glancing pedestrian, let's say, but given that we both have excitatory and inhibitory synapses plastic here, we're quite happy to have achieved this. And as far as I know, there are no other similar plasticity rules that allow such stable yet flexible tuning of synaptic receptive fields. Okay. Next party trick for codependent excitatory and inhibitory plasticity was for us to look at whether it has an impact on dendritic structure or input structure along dendritic trees. And so Everton, who, by the way, made the majority of this presentation for me, so that I wouldn't embarrass myself, thank you again, Everton, drew here a pyramidal neuron in 3 different colors. So the soma in black, and then proximal dendrites in this beige color, color that older people like to dress in, and then maybe more hip distal dendrites in orange. And of course, when you build a model of such a pyramidal neuron, you will build the dendrites as compartments, and we can actually look at just a distal compartment and a proximal compartment, as well as the somatic compartment. And what the immediate consequence of this is, when you put additional dendritic compartments onto a simple integrating fire neuron, is that the proximal compartments are still going to be bound by the threshold for action potential initiation, and so they can simply not wander far away from that threshold, at least into the depolarizing direction, because they will evoke action potentials and then be reset. And you can see this here, where we're looking at the membrane potential over 3 seconds for a proximal dendritic compartment. But the distal dendrite is quite uncoupled from the soma, and thus quite uncoupled from the somatic firing threshold, and will as such frequently wander into highly depolarized areas of this plot on the y-axis. So much higher than the threshold for firing. As a result of that, the currents for proximal dendrites that a proximal synapse can experience are much higher than the currents that the other way around. A current that a distal synapse will experience, it can be much higher than what a proximal synapse will experience, and we're plotting this here, where the membrane potential as experienced by a synapse is plotted on the x-axis, and the currents that will be experienced, NMDA and GABA currents, are plotted on the y-axis. And you see here that both the GABAergic currents, so the chloride currents, as well as NMDA currents, will go up in amplitude quite a lot as a function of the voltage Of course, at some point, the NMDA current must collapse because NMDA has a reversal around 0. So when 0 hertz are achieved, that NMDA current will go to 0. In this regime between -60 and -30, the NMDA current amplitude is largely dominated by whether the magnesium block is present or not, and so you will see a strengthening of the current as a function of the depolarization, maybe somewhat counterintuitively. So when you deliver excitatory and inhibitory currents to a distal dendritic compartment, as long as you're delivering both excitatory and inhibitory current that are in sync with each other, you get presumably or potentially strong LTP from the excitatory part of this, excitatory currents that are being delivered to the synapses, or to the vicinity of the synapses. But the gate for excitatory plasticity is essentially closed because equally strong inhibitory currents are also delivered. So you get a stable scenario in which synapses at the dendrites do not change much. The same is of course true also if you deliver weak currents. So if the synapses in the vicinity are all weak, as long as excitatory and inhibitory currents are in sync with each other, so locked in time and equally strong, then you will experience weak LTP from the fact that there's simply not very much excitatory current being delivered, and that is true even despite the fact that inhibitory currents would essentially allow for excitatory synaptic efficacy changes to happen based on this idea that weak inhibition will favor strengthening of excitation. Okay. When we imbalance excitatory and inhibitory currents, so when we deliver currents and the excitatory and inhibitory channels that are not necessarily synced with one another, then this will look slightly different in that, for example, if we deliver excitatory weak currents but strong inhibitory currents, we still don't see very much because the gate that inhibition is presenting is closed by way of strong inhibitory currents being delivered, and there is nothing to strengthen anyway because excitatory currents are weak. But when we do it the other way around, that excitatory currents are strong, inhibitory currents are weak at any one of these given membrane potentials in the distal dendrite, we see that now the inhibitory gate is no longer closed because there's not a lot of inhibition around, but this excitatory effects of the excitatory current being strong will lead to a potential, long-term potentiation of the excitatory synapses. Okay, so this has effects on the structure because if you are able to effectively control the membrane potential as a group of synapses in a coordinated way or in an uncoordinated way, this will imprint itself into the synaptic distribution of strengths. So if you start with a single dendrite here, to which you deliver lots of different stimuli, and you overrepresent some of these stimuli. So, for example, this green pathways, being represented through what looks like 70% maybe here of the synapses, and all the other channels, all the other inputs that are being delivered to this dendrite are delivered through the other 30% of the synapses. Then after learning, you will find that the uncorrelated signals have been sort of pushed out by way of the excitatory synapses of the correlated excitatory synapses strengthening thanks to their pretty good control of the membrane potential. And this, of course, works differently if you have only a minority. So if you deliver correlated inputs only through a minority of the number of available dendritic inputs into any given compartment, then they will, over time, actually weaken and be pushed out by the uncorrelated inputs. And one could make the point that depending on what the set point is, so depending on how many correlated or uncorrelated synapses you start out with on any given dendrite, you will have neurons that have single feature dendrites or mixed feature selectivity in their dendrites, so that in order to evoke action potentials from any given dendrite that is that is organized like this, you will need inputs from more than one sensory feature, whereas here you would only need a single sensory feature to evoke action potentials. What matters here in terms of determining whether it'll be a single feature or a mixed feature selectivity dendrite is the size of the correlated group, and we can plot this here. I apologize, these plots are not totally intuitive and we haven't quite worked out yet how to make them more intuitive, but what we're plotting on the x-axis is the size of the correlated group of synapses. So when this is 0, there's only a small fraction or a non-existent fraction of synapses that all receive the same inputs, whereas when this is 100, it means that virtually all the synapses onto that dendritic compartment are playing the same signal into the dendrite. And you see that the correlated synapses will have the majority of the excitatory weight. So what's on the y-axis here is the cumulative weight of these two different groups of synapses, the uncorrelated synapses or the correlated group synapses. So when you see a high value for the currents delivered through the synapses of the correlated group of synapses, that means that they have essentially taken on all the inputs and pushed out all the other inputs, whereas if you see high inputs currents from the uncorrelated groups, it means that the correlated group has been pushed out. And this happens only in this scenario when there is very few correlated signals synapses around, but as soon as you have more than 10 correlated synapses onto the same dendritic compartment, you find that the correlated synapses will hog all the synaptic weights of that available essentially for delivering synaptic inputs to that dendrites, as long as excitatory and inhibitory currents are not co-delivered. Because at that point, remember that whenever inhibition is equally strong as excitation, it will gate excitatory plasticity. And so this taking over of the dendritic structure by correlated inputs can only happen in a situation in which excitation and inhibition are not coordinated. When you match excitation and inhibition, the number of correlated synapses or coactive synapses you need in order to take over the selectivity of that dendritic branch is much, much higher, because essentially what happens is that inhibitory synapses cancel or gate off or disallow for any excitatory strengthening when excitation and inhibition are matched. And so in that situation, when you start out with matched excitation and inhibition, your dendritic compartment will end up being selective to many more different sensory features than just those of the correlated group. In the proximal dendritic compartments, the situation is the same for randomly delivered E and I currents. That's not surprising, again, because excitation and inhibition are not matched, so excitation has an easy time strengthening its synapses because it's not being gated off by excitatory, by inhibitory activity. And what's more, maybe it's essentially the same, but in distal synapses, in the matched scenario, the situation is quite different in that proximal synapses are, even when they're matched with inhibitory inputs, much more likely to evoke postsynaptic spikes, and so there's simply more pre-post pairs in proximal dendritic for proximal dendritic inputs than for distal dendritic inputs, and as a result of that, in the proximal dendritic compartments, you need much fewer synapses that are matched in their activity or correlated in their activity, excitatory synapses that are correlated in their activity to take over the entire dendritic structure and make it single feature detecting or single feature selective. And this is, again, what I just told you, just as a summary plot, where, again, the size of the correlated group is on the x-axis, and the distance of the dendritic compartment from the soma is plotted on the y-axis, and you see whether the correlated synapse group wins or the uncorrelated group synapse wins according to whether it's black for the uncorrelated synapse groups are dark or colored in for the correlated synapse groups, and you see that for random excitation inhibition, it's quite easy for the correlated activity to take over. When you have matched excitatory inhibitory inputs, you need a much larger group of correlated synapses to begin with to start dominating the output of that dendrite. Okay. I realize I'm flying through this talk, so I should probably talk a little bit slower for the last 10 minutes. But I guess I'll have a little bit more time for questions that way, and maybe it's not so bad to hang out in front of your computer late in the afternoon anyway. But apologies if I'm going too fast. It's a relatively straightforward story, I think. In this case, in terms of clustering, I should say we're again not the only ones who've proposed a rule that clusters synaptic inputs. Certainly, there's other research groups like Juliana Georgiev's group is looking at clustering in dendrites, and again, the voltage-dependent rule that Claudia has put forward also clusters. We're happy that ours can cluster as well, and that we seem to be able to explain quite a diverse set of experiments without having to retune our rules. And so this slide is simply a reminder that you see clustering in dendritic inputs. Of course, our rule can also explain why dendritic inputs shouldn't be clustered, and it actually may be able to contribute to the ongoing discussion as to whether pyramids do cluster their inputs after all. Although I think this debate is not nearly as hot anymore as it used to be about 10 or so years ago. In any case, that's for us another neat thing that codependent plasticity can do. And here is maybe the last bit of what codependent excitatory and inhibitory plasticity does for us, and this is what actually we started out looking for, because I've had a few publications together with Guillaume Hennequin back when I was a postdoc, but also when both Guillaume and I were working in England. We were looking to reproduce activity in motor cortex that is observed when a monkey makes a reaching movement from a resting or from a starting position to a targeted to a cue. When the monkey is being recorded from, you see that the neurons in motor cortex will display some kind of preparatory ramp activity that is in accordance with the target that the monkey is supposed to go to. So these red targets and green targets differ from one another in these red and green traces differ from one another in the cue that the monkey was told to reach towards. And then once the monkey starts initiating movement, you see a large diversion away from the baseline activity that this neural network has been puttering away at. That is distinct again for the motor movement that the monkey's making. So if it reaches towards a red cue, it will have a very different wiggle, I call them, than when the monkey reaches towards a green cue. And we were able to reproduce this kind of behavior in what we called stability optimized circuits. Those were rate networks, in which we had 500 or so linear neurons, or rate neurons, I should say, that interacted with each other via strong random excitation and then finely tuned inhibitory activity that was tuned by way of a synaptic plasticity rule, a non-activity dependent plasticity rule. And we find these stability optimized circuits quite nice because they seem to reproduce experimental data, but also because they give us an opportunity to sort of create computations by way of changing structure in a network and then ultimately getting consequently dynamics or change in dynamics out, and you can use these transient and amplified firing rate patterns to produce any kind of muscle movement or the equivalent in a computer model. And so we wondered whether we would be able to do something like that in spiking neural networks, and we really didn't have a good way to tune, especially inhibitory weights in spiking networks, but also we similarly didn't really know how to keep excitatory neural networks under control in the presence of a rule that aims to strengthen many of these excitatory connections, because that's what we knew we needed to create transient activity from the rate networks, in which we had to strengthen excitatory connections by tenfold from what we started out with. So we needed strong excitation and structured inhibition. And so we started out with a recurrent network. In this case, I think it was 1,250 neurons, 1,000 excitatory, 250 inhibitory neurons. And this network is delivering activity as expected from randomly connected networks with a correlation of EI that is already intrinsic to the system, a specific, effective time constant from the activity in those networks around 3 milliseconds, and a relatively constrained distribution of firing rates somewhere between 1 and 10 hertz. And you see here that we start out with weights that are relatively standard, let's say, in the literature. They're really not important other than that you should compare them to what happens after we turn on both excitatory and inhibitory synaptic plasticity, which is quite nice for us. It doesn't look particularly remarkable. So correlations of E and I have increased a little bit. The effective time constant has gone down by about to about 1/2 of what it was before. So this network got substantially faster, and the distribution of firing rates of each neuron has become substantially broader. So some neurons fire at much lower rates, some neurons fire at much higher rates, even if the average hasn't changed much. The firing rate, the weights that are coming out of this network after learning are somewhat interesting. They're sorted by their strength from excitatory to excitatory neurons, sort of that the strongest ones are numbered low and the lower, weakest ones are numbered here. And the inhibitory to excitatory weights behave similarly. Of course, they're not quite as nice because we sorted by the weights of the excitatory synapses, but you can see the same trend. In fact, those neurons that receive a lot of strong excitatory weights will also receive a lot of strong inhibitory weights. And that makes sense because the inhibitory weights are aiming to balance the network. Now, the output excitatory neurons of this neuron, and there's only excitatory outputs because we're obeying Delian rules, are inverse to that. So synapses of neurons with strong excitatory and inhibitory input weights tend to be weak on average, and synapses that are coming from synapses from neurons that receive weak excitatory and inhibitory weights will have strong connections outwards. So this was a little bit puzzling to us, and to be honest, we haven't fully understood why that should be a good idea in a network. But when we looked at the dynamics of the network, we got exactly what we wanted to get out of the network, which is a strongly amplifying, transiently active, but generally stable network. So I'm showing you this here by way of 3 examples that are each being presented with 5 different stimuli. And by 5 different stimuli, we simply mean we're driving 5 different patterns of activity into these 1250 neurons for the duration of the stimulus here. And you see that some of these will have relatively little effect. They might hyperpolarize the neuron during the stimulus, and there will be a depolarizing, there might be hyperpolarizing, I should say, a decrease in rate during the stimulus, and then a slight increase in rate during the aftermath, or in the very few seconds after the stimulus is shut off. And this might be the case for all of the inputs for some neurons, but for some other neurons, they will have a very strong response already during the presentation of a stimulus, and other neurons will have no response during the stimulus presentation, but once the stimulus is being shut off, these neurons will respond with a very strong off response to that stimulus, seen here in these raster plots, but of course also seen in the average activity. And if we average the firing rates for all the neurons, we see that, again, some of these neurons have a really strong amplifying response away from the baseline. Of course, you know, when you're averaging over many, many neurons, this doesn't look particularly strong, but keep in mind that this is really the average firing rate response of over 1,000 neurons, and they, on average, respond for some stimuli with a 5 Hz stimulus after the stimulus is shut off. So we were quite happy about this because it meant that we were able to reproduce these stability optimized circuits that we had produced in linear networks, and basically deliver a proof of principle that synaptic plasticity rules should be able to create networks that are naturally amplifying. And we're now in the process of learning sequences into these networks where we basically try to teach the networks to play a little tennis for us, but we're not quite sure yet how. So it turns out that the dynamics that we observe inside the network are quite stubborn towards our efforts to modulate them in a targeted way. So they like to do what they want to do, but not what we want to do. And we might have to go to the level of the readout neurons later on to get different kinds of activity from them. And with that, I'm sorry to report, I'm pretty much done. So the summary here is that we built a set of rules, excitatory and inhibitory synaptic plasticity rules, that always play nice with their friends. That means that they coordinate, so excitatory plasticity pays or heeds not just to pre- and post-synaptic activity from its neurons, but also pays attention to the currents that are delivered to the vicinity of that synapse. And similarly, inhibition does the same. The result is rules that allow us to co-tune excitation inhibition effortlessly, that create stable networks with structured dendritic inputs according to initial starting structure, and that also allow us to build networks and spiking networks that are inherently stable, so they cannot go out of bounds, but they still produce transient amplifying dynamics that we can allow for, that we can use for population computation or any neural processing, really. This is quite a nice starting point to build spiking networks that have functions beyond, let's say, the most simple memory engrams, and we're looking forward to working with spiking networks for a little while longer. I should say, like several other learning rules, we've been able to fit a surprising number of experimental findings, and maybe, of course, when you're a hammer, the world looks like nails. Of course, we think that our learning rule is vastly superior to any other plasticity rules out there. And of course, the proof of the pudding is in the eating. So we'll have to see whether our hubris with regard to the superiority of our model will hold up, or whether it will be washed down with a cold glass of water delivered by our favorite experimentalists, hopefully. So with that, I should thank again Everton Agnes, who's in Basel, and also the other members of my group. This was a very recent picture. You can see we're all hiding our masks, but Austria is actually currently out of lockdown. So we're in the lab, and we can even transiently come together for a quick picture before we all run apart again. But it's been lovely moving to Austria. I've only arrived here in August, and it's a really lovely place to work. So if you're looking for a position, a PhD position, or a postdoc position, I can highly recommend you look into your options at IST Austria. So with that, I guess I'll stop my slideshow, and I'll be able to look at all the comments that have been rained upon me, hopefully not. Thanks again for the invitation. I hope I didn't bore you too much, and please do let me know if you have questions.
Thank you so much, Dr. Vogels, for the very interesting talk. So we will now transition into our Q&A session. I was thinking I'm going to invite Everton online. He is welcome to decline the invitation, but it might be a nice discussion to have him with us on screen for this. And then I will also bring Siobhan on, so she can start with the other questions that have been posted so long. And also to all the other audience members, please do remember to post your questions. Then we can ask them either on your behalf, or we can bring you on screen for you to ask it yourself. Hi, Everton.
Hello. I'll just be around here.
You'll just make sure that I don't say anything silly. I'll let you answer all the questions now that you're here. So this is Everton. Very happy that you're here, Everton.
Siobhan, I'm handing over to you now.
Okay. Does Dean not want to come on to ask his question?
Let's see.
I can hop off again.
Okay, cool.
I think
Dean,
you
maybe
can have
give
up
us
to
the
6 people.
invitation.
I think if you just invite people blindly, they usually come on if they feel comfortable. And you can have up to 6 people on screen, so you don't have to even leave, at least.
Yeah, that'd be great.
Okay, let's see if we bring Dean on. Yes, our plan is limited to 4 people on screen.
Oh, really? Oh, no.
Okay, the Dean is
Come to Worldwide Neuro.
There we go.
Hi, Dean. Welcome.
Hi.
Hey, Dean.
Okay, so Tim or Everton, I was wondering what kind of distributions of synaptic strengths arise under this rule. Like, do you get unimodal distributions? Do you get heavy tails? Does it allow for strong bidirectional connections?
I think they're mostly heavy tails. Yeah, Everton? Yes.
Sorry, my mic was mute. Yeah, in the recurrent network, when it like evolves naturally, it becomes heavy-tailed, and that's one of the reasons it would give this amplification, because it's non-normal, right? So you have some propagation of activity. Okay, thanks.
Cool.
Like, I wish I had more questions, but that's the only one that popped up for me.
Cool. Then we'll move on to the next question. Thanks, Dean.
Sure.
Okay, let's see. So, Christoph, I will invite you on screen as well. Please feel free to decline if you don't want to come online.
Wait another 10 years, maybe 15. Be wearing beige in no time.
Hi.
Hello, Christoph.
Very nice talk. I was wondering about if the nearby excitatory and inhibitory synapses affect the plasticity rules instantaneously, or if you have a certain time scale there.
Again, Everton, correct me if I'm wrong, but I think there is a natural time filter by the way that they're acting like low pass filters, essentially. And so the currents are not immediate. Like conductances will evoke currents, and then you have an effective delay just by it being conductance-based synapses. Right, Everton?
Yes, so there is no pass filter from the currents, so that the actual variables in the plasticity model is filtered version of the currents. So there is a natural delay from that. And also to compensate, because like I used in the simulations GABA A and MDA, so they are roughly 10-fold apart in time scale. So this you need some structure there, and this is used to fit the data then too. I mean, this time scale is somehow important there.
Okay, because I could imagine.
Yeah, go ahead.
I could imagine that depending on how far away your synapses are, right, it takes a bit more time than to influence. But you're not really looking at this, right? I mean,
so we've looked at it in dendritic compartments, but only like either we look at it in a single compartment where there is no distance, right, or we look at synapses in one of 2 or 3 compartments. But so there's no direct distance effect that is analog, right? It's either you're in the dendritic compartment or not. But yeah, you're right. I mean, also currents attenuate, right, over distances. So there's an additional spatial filter in there.
I could also imagine that, I mean, you showed the runaway activity of excitatory and inhibitory activity, right? If you don't have this codependence, probably would depend on the timescales, right?
Yeah.
How fast this? I was just wondering if you also tested for this. Or
you mean experimentally or in models?
I mean, in the model, if you
yeah, for sure, in the model. I mean, yeah, we mostly want to. Yeah, if you're the reviewer, we'll do it for you.
Okay, cool. Thanks.
Thanks, Christoph. Ashman, I'm inviting you on screen now. Okay, I'm going to try and invite Prof. JS as well.
I can answer the question regarding sleep. So these experiments were run mostly while Everton was sleeping, but we didn't particularly model wake and sleep cycles in that our neurons don't have different activity states. So yeah, no, sleep is not in the budget yet.
Perfect, thank you. Okay. Welcome, Prof. JS.
Oh. Hi, hello. Very nice presentation, Tim and Everton. Very nice work.
Yeah, Everton made the presentation. I only said the words that he told me to say.
Still, I was thinking this is something similar to the previous question from Christopher. But I have the impression that this codependent plasticity is going to be it's going to be more effective when the synaptic contacts are really close physically. So I was wondering whether I mean you have seen the experimental evidence, you have a much better idea than just my intuition. But I was wondering if you would expect to find similar effects when you look at different structures. I'm thinking, for example, instead of proximal or distant apical dendrites like you showed in the model, could we have something similar if we look at, for example, two basal dendrites that are technically different, but they might be close to each other?
Yeah, so the thing is, I would imagine that the currents will not affect much. Then the interaction will be indirectly, right? Because what could happen if the two basal dendrites then connect to the soma, but they don't interact directly, the activity of the soma or the spike train would be what's driving somehow plasticity in these dendrites, right? So whether one dendrite correlates more with the postsynaptic activity or vice versa, then these types of competitions could emerge. But I would think that this is more indirectly because of this physical separation, as you said, right?
Okay. Yeah. And
there is some... Go ahead.
Yeah, sorry.
No, go ahead.
No, I was thinking, I mean, same thing could be said, for example, if we look at different branches of an apical dendrite, some more second order ramifications or something like that. So it was my intuition, but I wanted to make sure that it really is especially effective in close proximity of the contact points, right?
Experimentally, yes. Then in the model we try, I mean, as Tim said, we separate in compartments, right? So we don't have the distance per se. This is a bit more, requires a bit more work in the modeling part to implement all this kind of structure and plasticity on top of that for the whole dendritic tree. So we divide in compartments, and each compartment has its, let's say, pool, right, of excitatory and inhibitory currents that it sees. But of course, now if the signal is propagating from far away, you have all these other effects, right? So you have the indirect effects that would come from that, which is how the signal propagates, how the voltage is fluctuating, giving different compartments, right? Which we will also have in the simulation, but then this is the classical effect of how correlated different inputs are in the dendritic arbor.
Okay, thanks a lot.
I concur.
Perfect, thank you. Okay, any last minute questions? I'll bring Siobhan back on.
Can also leave the video.
Oh no, you're welcome to stay. You've got an extra screen now.
Since you put on a shirt, Everton, last time in 4 weeks someone sees your shirt.
Yeah. Now it's a few opportunities to. There's a shirt at home.
You were just cut out.
Cool. Yeah, I think let's give it a couple more minutes just to see if anything else is coming in.
You can always send me a text or send me a message on Twitter or whatever.
Yeah, I think we can round it out. Virtual computers.
Thank you for the opportunity.
Yeah, thank you so much. And thank you, Everton, for joining us on screen as well.
Definitely.
Then I will just share my final slide to round it out. There we go. Perfect. So yes, can everyone please give Tim Vogels a virtual round of applause in the comments for this very cool presentation. And thank you to all of you for joining. And then if you like this and you're interested to see more of NERV online, please do subscribe to our mailing list and our calendar. Siobhan will share those links for you guys. There we go, in the comments. Yeah, thank you so much. It was a wonderful opportunity.
Lovely. See you soon.
Bye, everyone.
See you soon. Bye, everyone.
Bye.